Our last topic for PowerPoint 17A is over microscopic anatomy. Cardiac muscle cells are striated like skeletal muscle cells, but their structure does differ. Cardiac muscle cells are short, branched, fat, and interconnected. They only have one central nucleus and many mitochondria, so that makes them resistant to fatigue. They have Z discs and A bands and I bands, all terminology present and also talked about in Chapter 9 of your lecture textbook. Chapter 9 of your lecture textbook use skeletal muscle as a model, and so we will be comparing cardiac muscle to skeletal muscle for that reason. The T-tubules in cardiac muscle are wider, so less numerous than that found in skeletal muscle. We find that the T-tubules for cardiac muscle only enter the cell once at a Z-disc. So we don't see the same type of association of T-tubules with sarcoplasmic reticulum, and the simpler sarcoplasmic reticulum of cardiac muscle cells is not going to lend itself to the formation of triads. Intercalated discs are going to be connecting junctions between cardiac muscle cells, and they contain desmosomes and gap junctions. The desmosomes hold the cardiac muscle cells together, and the gap junctions allow ions that are electrically coupled to pass from cell to cell. The gap junctions are allowing the heart to beat at once or as a coordinated unit, and this is called a functional syncytium. Whereas the desmosomes are not just holding cells together, but they actually prevent the cells from separating during the contraction of the cardiac muscle cells. There is give, a little bit of stretch allowed with the desmosomes. We also have desmosomes in our epithelial tissue. So if you look down at your arm, that tissue you're looking at contains desmosomes. So if you have an itch and you scratch it, we don't see that our epidermis just shears or kind of tears off our skin because we allow for that little bit of stretch for everything that both our epidermis and heart has to encounter or endure throughout its function. We also want to talk just briefly about the intercellular space between cells. It has a connective tissue matrix called endomysium. Now you can kind of compare this to the cardiac skeleton of the myocardium because it does connect the cardiac muscle to the cardiac skeleton, this endomysium or tissue matrix. And so it's definitely a part of kind of that middle layer of the heart. But we have to look at it more on a microscopic level and look at how this fine tissue membrane is helping connect these two myocardial parts. It really gives the cardiac muscle cells something to pull against. Again, we're looking at a figure from your lecture textbook. So figure 17.11a is showing the microscopic anatomy of the cardiac muscle in this image. And this image is showing you not only the striations of cardiac muscle cells, the uninucleate nature, but also the interconnections called intercalated discs that we find between our short branch cardiac muscle cells. If we take a closer look at these intercalated discs, we would see that they contain these hollow gap junctions that electrically connect myocytes and desmosomes, which keep myocytes from pulling apart, but they allow for a little bit of stretch. You can think of desmosomes kind of like molecular zippers. There's a little bit of tension or give on each side of their interconnecting cadherin proteins. And here is figure 17.11b, which is just showing you kind of an overall depiction of what a cardiac muscle cell would look like. We can see here is an interconnection between one cardiac muscle cell and another. There's branching seen on both cells. If we kind of do a cross-section of the cells, we can see the numerous mitochondria. We can see the uninucleate nature. We can see the blue sarcoplasmic reticulum, some T-tubules, and we can see that cardiac muscle cells also include myofibrils, which include the myofilaments, actin, and myosin. And those myofilaments are arranged in a particular manner as to create the dark A bands and the light I bands. So let's continue on and compare skeletal and cardiac muscle. So there's some similarities that cardiac muscle has with skeletal muscle, and that is that muscle contraction is preceded by a depolarizing action potential. So an action potential is going to occur before we get that muscle cell contraction, and that the depolarization wave does get from the sarcolemma of the cardiac muscle cell into the cell via the T-tubule. And that T-tubule carrying the action potential, that depolarizing wave into the cell, will cause the sarcoplasmic reticulum, or SR, to release calcium. And when calcium is released, we'll see intracellular calcium levels rise and trigger the excitation-contraction coupling. The excitation-contraction coupling step is going to see calcium bind to troponin, move tropomyosin out of the way, and lead to cross-bridge formation. So those are some similarities. And if a lot of that information seemed new to you or you forgot it, please go back and review Chapter 9 in your lecture textbook. There's also some differences, however, and these we do want to highlight. And that would be that cardiac muscle cells can be self-excitable, and we didn't see that for skeletal muscle cells. Now, not all cardiac muscle cells are self-excitable, but some of them are. So there's actually two kinds of cardiac muscle cells, or myocytes. There are contractile cells, which are pretty much like general cardiac muscle cells. They're responsible for contraction. When activated, they contract. But we have these specialized self-excitable pacemaker cells in the heart as well. And these are non-contractile cells. They don't really contract. They're not responsible for contraction of the heart. But they are responsible for spontaneously depolarizing, for initiating the action potential that we're calling a depolarizing wave that is sent through gap junctions throughout the entire heart and ensures functional syncytium. These pacemaker cells do not need the nervous system in order to be stimulated, in order to open channels and cause a depolarization. In fact, they spontaneously, so by themselves, depolarize. And that's a huge contrast to skeletal muscle fibers, which required a motor neuron in order to initiate the original depolarization that traveled throughout the entirety of the sarcolemma in skeletal muscle. So big difference there for skeletal versus cardiac. Cardiac muscle cells have pacemaker cells that are non-contractile and start the depolarizing wave in the heart. So the heart does contract as a unit. We have these pacemaker cells, they kind of start the signal, but this signal goes to the contractile cells, so all cardiomyocytes, via the gap junctions. These gap junctions at interarticulated discs are connecting cardiac myocytes, and that's going to mean that all cardiac myocytes contract together or none at all. So this is going to ensure an effective pumping action. Skeletal muscles, on the other hand, they're acting independently. We also saw with skeletal muscle that our calcium influx or increase in the cytoplasm, so within the sarcolemma or plasma membrane, it all came from the sarcoplasmic reticulum. All that calcium was released from the sarcoplasmic reticulum in skeletal muscle. But in cardiac muscle, we're going to see the slow opening of calcium channels after this initial depolarization. And these calcium channels that are slow opening happen to be in the sarcolemma. And the reason they're opening is they're actually allowing extracellular calcium into the cardiac muscle cells. So for cardiac muscle cells, we're getting calcium from outside the cell, so it's extracellular, and from inside the cell once it's released from the sarcoplasmic reticulum. It's actually the extracellular calcium ions that cause the sarcoplasmic reticulum to release the intracellular calcium ions. Whereas skeletal muscle, it doesn't use extracellular calcium ions. We do this because, or I shouldn't say we, I should say cardiac muscle does this because we want to prevent tetanic contractions in cardiac muscle. And this works very well. Tetanic contractions cannot occur in cardiac muscle. And what tetanic contractions are is a tetanized state or kind of a physiological state. The latter is really kind of differentiate from the disease tetanus. It's a sustained muscle contraction that's evoked when the motor nerve that innervates a skeletal muscle emits action potentials at a very high rate. So skeletal muscle there is mentioned because this isn't occurring in cardiac muscle. So if I kind of break that down a little bit, what we're saying is we do not want cardiac muscle cells to be able to have a sustained contraction. We want them to contract all at once as a unit, but then relax because we want to ensure a heartbeat. We want to ensure the pumping action of the ventricles. So what we can do is by increasing the amount of calcium that's going into cardiac muscle cells is we can prolong the action potential. Therefore, we're prolonging the absolute refractory period or the period of time during which another action potential cannot occur. So we have a much longer refractory period in cardiac muscle cells than skeletal muscle fibers. And this absolute refractory period is almost as long as the contraction itself, and that's what is preventing the tetanic contractions. We get a prolonged contraction, but once it's over, and then the heart is allowed to relax and fill as needed in order to be an efficient pump. The heart also relies almost exclusively on aerobic respiration, and cardiac muscle has more mitochondria than skeletal muscle, which we established, And that means they have a greater dependence on oxygen. Because what mitochondria do is carry out a process called cellular respiration. And in order to carry out this process called cellular respiration, they need oxygen. And this process that uses oxygen creates ATP. And energy is important for the heart. And so cardiac muscle cells cannot function without oxygen. Skeletal muscle actually can go through a process called fermentation, where it can make ATP even when oxygen is not present. Whereas cardiac muscle doesn't do that. It actually would rather use other types of fuels besides glucose, things like lactic acid or even broken down proteins in order to make sure that they're able to produce ATP. It's able to use adaptable fuels, but not function in a condition where there's no oxygen. And that's it right now for kind of the microscopic anatomy. I do recommend looking at table 17.1. We want to be able to compare on the test differences between skeletal and cardiac muscle and recognize what belongs with which one. So for example, both skeletal and cardiac muscle cells are striated, cardiac muscle cells are branched, and whereas skeletal muscle cells are multinucleate, cardiac muscle cells are generally uninucleate, at most would only have 2 nuclei. We only saw the intercalated discs in cardiac muscle cells, or between them, I should say, and those cardiac muscle cells would have in their intercalated discs gap junctions and desmosomes. It's really important that you know which type of muscle has intercalated discs, what type of cell junctions are found in intercalated discs, and what each one does. Very much could be a question on your test. We also know that T tubules, A bands, I bands, Z discs, even the myofilaments, actin and myosin, are found in both skeletal muscle and cardiac muscle. But the sources of calcium did vary. We only got sarcoplasmic reticulum sources for skeletal muscle, whereas we had sarcoplasmic reticulum and extracellular fluid sources of calcium for cardiac muscle. In both, calcium binds to troponin, but only in cardiac muscle were pacemaker cells, which were non-contractile, present, which ensured functional synctium. And last but not least, the supply of ATP was only aerobic in cardiac muscle cells. They had more mitochondria and they rely on oxygen. They were fine with using other types of fuel besides glucose, but they were not fine with not having oxygen. Whereas aerobic and anaerobic respiration can occur in skeletal muscle cells. So the supply of ATP differs just a little bit. So make sure to go back and review the microscopic anatomy. Check out chapter 9 in your lecture textbook if you need a deeper review. And then stay tuned for part B of chapter 17.